Hello, everyone. My name is Matt Drayluck, and I'm presenting my master's level research currently in progress on the effects of long-term exposure to multiple stressors on the biochemical pathways of the blue mussel. Here's where I grew up, as an intertidal creature myself. Here's me trying to demonstrate some early ecosystem engineering skills. I made lots of friends as a kid, like this crab that I temporarily adopted. And a lot of years may have passed since that wedding, but well, mostly nothing's changed for me. Currently study the sessile marine invertebrates in the highly diversified habitat of the rocky intertidal zone. Creatures in this area exhibit a wide range of tolerance to multiple biotic and abiotic stressors, while tidal and seasonal conditions change the environment around them constantly. These tolerances are being strained further by anthropogenic climate change, with documented environmental impacts on intertidal ecology and aquaculture on ecosystem scales. Many studies have illustrated that environmental stress can have impacts on individuals' physiology and biochemical processes. Most intertidal species already have a limited ability to increase their thermal tolerance strategies above their current ecological conditions, meaning they're living on the edge already. Many studies have focused on modeling the direct abiotic responses of temperature, showing changes in distribution or abundance. However, studies of biotic stressors such as food availability or predation risk have also shown significant impacts, and including them in our predictions captures a more complex picture with higher resolution than temperature alone. In this study, I aim to understand how the interacting effects of long-term exposure to abiotic and biotic stressors drive biochemical processes and impact physiological performance at the subcellular level. I'll do this by exploring the impacts of 3 experimental stressors. 1, warming ambient waters. 2, decreased food availability. And 3, non-consumptive predation risk, or the feeling you might be eaten by a dog whelk. An ideal place to start modeling future impacts of climate change is where we've observed them already. The waters in the Gulf of Maine have sea surface temperatures that have increased more rapidly than 99% of the rest of Earth's oceans. Many studies also show declining populations of mussels, Atlantic cod, and American lobster in the last few years. So let's go back to my friends literally stuck in the intertidal zone of the North Atlantic Ocean. The blue mussel inhabits the lower to mid-intertidal zone all around the North Atlantic. The blue mussel experiences a wide variety of environmental stressors on a daily basis due to the vertical gradient of the intertidal zone. Edulis is a food source for other predatory organisms, as well as an economically valuable aquaculture species. So we're going to use it in this project specifically in a multifactorial exposure design to measure the subcellular response. There are 2 questions I plan to address. How do these stressors impact both anaerobic and aerobic pathways in the production of energy? And 2, are mussels able to store glycogen when experiencing long-term starvation and other stressors simultaneously? Let's explore the exposure design from here. 240 Edulis were collected off the north coast of Nahant, Maine, and acclimated in ambient seawater tanks for a week at Northeastern University. Mussels were equally divided into 8 treatment groups based on 3 chosen stressors, as diagrammed by the figure on the right. Then mussels were collected and sacrificed at different time intervals over the course of 12 weeks. At Sonoma State University, the subcellular effects have begun to be analyzed. Protein extracts from adductor tissue are being used to assess aerobic and anaerobic metabolism functioning. I am performing assays to determine activities of 2 key enzymes. First, citrate synthase, the rate-limiting enzyme in the TCA cycle, to represent how actively mussels are aerobically respiring within their cells. Secondly, to assess anaerobic metabolism, cytosolic malate dehydrogenase will be assayed for activity to infer energy allocation strategies between aerobic and anaerobic conditions. I do believe that these stressors, over the long term, will impact the flux between anaerobic pathways and aerobic pathways from control levels. I expect increased water temperature to drive higher activity in both enzymes due to simple reaction kinetics with unregulated body temperature. Decreased food availability will increase aerobic activity as they break down their glycogen stores to use a more efficient pathway than the anaerobic glycolysis. Predation risk will cause mussels to close their gape as a defense mechanism, meaning they may reduce the flow of water over the gills and kind of hold their breath. But I suspect that as the experiment goes on, they cannot sustain this and they may run out of glycogen. Secondly, assessing the impact of these stressors on energy storage. Temperature drives filtration rate in mussels, so I expect that temperature alone should allow them to pack away nutrients, including glycogen. Similarly, loss of food availability should decrease their glycogen stores, as starvation will leave mussels no choice but to use their reserves. I expect that the stress of non-consumptive predation risk will also lower stores as CMDH activity and glycolysis increase, burning through those reserves. In conclusion, what the data for both of these questions will soon tell us is if interactive effects of these stressors may mask each other, resulting in antagonistic effects, or result in synergistic effects with cumulative multiplicative results higher or lower than the additive effects of individual stressors alone. Assessing organismal performance in response to wildly fluctuating and stressful environments can hopefully help us inform better predictions of responses to anthropogenic climate change. Thank you.